Pollen grain is also called male gametophyte. Gametophyte bears gametes. So pollen grain will have male gametes in it. So that's why pollen grain is also called male gametophyte. Now discussing the structure of pollen grain. Pollen grain is made up of two layers, outermost XII and inner one intine. You can see XII is not continuous throughout. At one point, the XII is absent, and the point where it is absent, that point is called germ pore. And through this germ pore, pollen tube growth will occur. Then pollen grain is having two cells, bigger one vegetative cell and smaller one generative cell. Now let's discuss about the chemical composition of the two wall layers. XII, it is the hard outer layer and it is made up of a chemical called sporopollenin. This chemical is most resistant organic material ever known. It is highly resistant to high temperature, strong acids, alkali and microbial attack. Even the enzymes cannot decompose sporopollenin. So that is the reason pollen grains are well preserved as fossils, because no enzyme can decompose pollen grain. So you can see sporopollenin or the XII is absent at a point, and that point is called germ pore. From that germ pore, pollen tube growth occurs. In monocots, there is one germ pore, and in dicots, there can be up to 3 germ pores. And through each germ pore, pollen tube growth can occur. Now the chemical composition of intine. Intine is made up of cellulose and pectin. Now talking about the characteristics of vegetative cell and the generative cell. Vegetative cell is bigger. It has abundant food reserve. This food reserve will be used during the pollen tube growth. Then it has large irregular nucleus. Generative cell is smaller. It is floating in the cytoplasm of vegetative cell. Generative cell is spindle shape with dense cytoplasm and a nucleus. Now, in 60% of angiosperms, pollen grains are released from the anther at two-celled stage. Those two cells are vegetative cell and generative cell. And in rest of the 40% angiosperms, pollen grains are released from the anther at three-celled stage. So before the release of the pollen grain, the generative cell in such cases divided into two male gametes. So such pollen grains have 3 cells, vegetative cell and 2 male gametes. Some people are allergic to pollen grains that result in respiratory disorders in them like asthma, bronchitis. You might have seen this grass growing wildly around and having these small white flowers. This grass is called carrot grass, and it is also allergic to people. Carrot grass is also called Parthenium. Actually, it is not the native of India. It was accidentally brought into the India with the imported wheat. And this pollen grains of this carrot grass are also allergic to people. There is some goodness in pollen grains also. They are rich in nutrients. So in the market, they are available in the form of pollen tablets and syrups. When Race horses and athletes consume pollen tablets or syrups. Their performance increases many times. Viability of pollen grains. The period for which the pollen grain is living or surviving. The period within which the pollen grain can germinate, that is called viability. Viability also depends upon the temperature and humidity. Pollen grains of plants like rice and wheat lose their viability within 30 minutes of their release. If pollen grains of such plants do not find right type of stigma within 30 minutes, they will fail to germinate. Then pollen grains of members Rosaceae, Leguminosae and Solanaceae, their pollen grains can be viable for months also. We can preserve pollen grains in liquid nitrogen at minus 196 degrees C, and this process is called cryopreservation. Now, why do we need to preserve pollen grains? These preserved pollen grains can later be used in crop breeding programs. Crop breeding programs means crop improvement programs. We can use these preserved pollen grains to produce hybrid improved and better yielding varieties of crops.